Swayam Prabha. Digital India. Educated India.
Hello, welcome to Module 2, Week 1 of our course on Introduction to Dynamical Models in Biology. In this module, we'll discuss about essential features or basic concepts that is required before you start making a model of a biological system. So let us start with some basic concept. While making a model, we have to keep in mind that no model is actually exactly same as the reality. Every model you make is actually simplification of a real phenomena. And the second key issue that we have to keep in mind is that as every model is a simplified version of a real process or real phenomena, it cannot answer each and every question. So every model can only answer a specific set of questions that you ask to the model. Let me explain this with a simple example. Suppose I want to give you the direction from airport to our institute, and I'm drawing a map on a piece of paper. So obviously, I will not draw each road and bylanes of the city. I will not show each and every house, shops all around in this road from airport to our institute. What I will do, I will only draw the main street through which you should reach our place, and also some important places which you can use as a marker or flag. So the map that I am drawing on a piece of paper is actually a partial representation of a reality, and it is almost like a model of our city. And the question that it can answer is, it can only tell you that how and through which route you should reach our institute from the airport. If you ask the map how to go to the stadium from airport, obviously that map will not be able to answer that question. So while making a mathematical model of a biological phenomena, you have to keep in mind that the model that we will make will be a simplified version of the reality or the real phenomena or the real biological process that you are trying to model, and you can only ask a specific set of questions to it so that it will be able to answer. So let us look into the steps in mathematical modeling, particularly dynamical modeling in biological systems. The first and foremost requirement is that you have to collect information. Information about the process that you want to model. Suppose you want to model the dynamics of signaling by insulin in our cells. So you have to collect all the information required for modeling this. So you have to collect information about the molecules involved in that signal transduction pathway. You have to know the processes involved. So where do you get that information? You get that information from published literature, from other people's model, maybe from some databases, or maybe you will be doing some experiments yourself to generate some data which can be incorporated into your mathematical model. Once you have collected all this information, you try to make some sort of simplifying assumptions. You make some assumptions so that your model becomes simplified. We will discuss about this simplification and assumptions when we will discuss about specific mathematical model in the subsequent modules. Once you have made those assumptions, the next third steps comes, you make graphical model. This is where you make a visual representation of the problem that you want to sort out through mathematical modeling. I will discuss about this graphical model in details in a couple of slides. Once you have made the graphical model, the fourth step is making the mathematical model that you essentially want to create. This mathematical model will be based on the information that you have collected at the beginning, then based on the assumptions you have made, and obviously the graphical model will help you to create those mathematical models. Remember, mathematical models are nothing but a set of equations that you write. And we will discuss in details how to write down those equations. Last part of mathematical modeling, particularly dynamical modeling in biology, would be analyzing that model. So let us start with graphical model. It is not essential that for every modeling you create a graphical model. But usually I advise that you try to create a graphical model first so that you will not miss any particular property, any particular process, or any particular molecules or elements involved in the process while making the mathematical model. Essentially, graphical model will be a map based on which you will write down the equations for your mathematical model. And graphical models are not something new. As a biology students, we are all very much accustomed with graphical model. Most of your textbooks, starting from biochemistry textbooks to molecular biology textbook, you will find this type of graphical model. For example, I have shown one model here, a signal transaction pathway model. This type of model, graphical representation, are very common in biology. We have to remember, most of the biological processes are very complex. Explaining that by word to someone, by text to someone, sometimes is very difficult. It is always easy if we can make a plot or a diagram or a network map, something graphical, which communicate the information properly to the other person. The same thing is here. You make a simplified visual representation of the complex biological process that will help you to create the mathematical model. So when you are creating mathematical graphical models, we should try to follow some consistent rules for that. Otherwise, we may miss some critical issues in the model. First thing, the first critical point is that you define the boundary and compartment of the system that you are dealing with. For example, see this example diagram I have drawn here. Suppose I want to simulate or model and simulate a process which involves a particular process in a cell. So a cell can be represented by an external boundary like this, as I have drawn by the bigger circle. Inside that, I have shown the nucleus. What I am trying to model here? I want to model the process by which a molecule A get transported from outside into the cytoplasm, and then from cytoplasm it enters into the nucleus. So in this case, I have to show three different compartments. The first compartment is the outside the cell. Second compartment is my cytoplasm. And the third compartment is nucleus. In this particular case, I have to show nucleus separately because the molecule is entering in the nucleus from cytoplasm. But in some other cases, you may not have to show nucleus as a separate compartment because there is no particular process going on nucleus which you want to model. So in that case, you have not to show nucleus as a separate compartment. So you have to judiciously identify which are the compartments involved in your process and what is the boundary of them. The second point is that you have to identify all the players involved in the process that you want to model. For example, in this case, in this example that I am drawn here as a graphical model, the molecule is present initially outside, as I have marked is that O, means A, molecule A, present outside the cell. It enters, I have marked that same molecule when it is in cytoplasm as AC, to remind you that this is the A molecule, but it is present in cytoplasm. The same molecule, when enters into the nucleus, I am marking that as AN, representing that it is the A molecule in nucleus. This type of naming the same molecule by different name when they are in different compartment has a particular advantage. Because now I can count and keep a track how many A are present outside the cell, because I will record the number or the values of that as the value for AO. I can also keep a record of how many AC are there, that means how many A molecules are in cytoplasm, and I can separately record how many AN are there inside the nucleus, that means how many A are present inside the nucleus. So note the crucial issue here. Although A present outside the cell, inside the cytoplasm and in the nucleus are same molecule, we have named them separately because they are present in 3 different compartments. Now suppose AN comes back to cytoplasm. This molecule which comes up to back to cytoplasm will not be marked as a new molecule, but rather it will be same as the AC that we have already drawn here. So in essence, what I want to communicate is that once you have decided the boundary and the compartment, then you start showing all the players involved in them and mark each of the molecules or elements or component present in each of these compartment uniquely so that you can keep a track of them. Then comes the third point. Till now, we have represented compartments, we have represented molecules or elements or player in the process. Now, we want to represent molecular events or processes. In most of the biological problems that we will deal with, we will have certain common type of processes involved, and I have only shown some of them in this slide. Does that not mean that it is an exhaustive list, but I want to give you an example that how we consistently draw certain molecular processes. The first one is simple reaction. A is becoming B. So you just represent that by A to B by an arrow, with an arrowhead in the product. Suppose, just like the previous example, A is present outside, so I have marked as AO. That is going inside the cytoplasm, so it is AC. This is just transport process. I can represent the transport process also by an arrow, just shown here. So AO is getting transported to AC in the cytoplasm. Many chemical reactions in biological system will be actually reversible reaction, the way I have shown here. A plus B making a complex AB, and it's reversible. That means A plus B will form AB, also AB will break down into A plus B. As you know that many reactions in biology are enzyme catalyzed. For example, see the example given here. A is becoming phosphorylated by a kinase. So this is my forward reaction. A is becoming AP, phosphorylated. Who is controlling that? K, the kinase is controlling that. Notice how I have written K. I have not written A plus K. I am not writing A plus K because in this process K is an enzyme, and by this process of phosphorylation of A, K enzyme does not get used up. Remember your basic biochemistry knowledge that an enzyme or a catalyst does not get used up in a enzymatic reaction. It only controls or modifies the rate of the process. So I am not showing here it as A plus K, rather I am showing that A is becoming AP and that is controlled by K as represented here by a arrow. Similar thing for a phosphatase. Once your A has become phosphorylated and become AP, it can become dephosphorylated by a phosphatase enzyme. Again, P is representing here the phosphatase, and it is controlling the reversible reaction. So remember what I am doing here. I am representing the control or modifier or the controller of this reaction by these two enzymes K and P, and they are drawn as a vertical arrows on the reaction that is controlled by these individual molecules. Now, here in this example, AP is getting dephosphorylated to A. So that means P phosphatase, which is removing this phosphate group from A, is actually working opposite to the kinase reaction, phosphorylation. So sometimes some people may want to represent it as if P is a molecule or enzyme which is inhibiting phosphorylation of A. So that can be represented here as I have shown, A to AP, this is the normal process by which A get phosphorylated to AP, and P is inhibiting it. Notice that we don't have an arrowhead here, we have a hammerhead. Usually this type of hammerhead represent inhibition. So what I want to represent here? I want to represent that P is inhibiting the phosphorylation of A to AP. So in brief gist, you use an arrow to represent activation or a reaction or transport, whereas you use a hammerhead to represent inhibition. We are drawing these unit processes. We will club these unit processes like a reaction, reversible reaction, inhibition, controlled by enzymes to create a larger map of particular process. Let us see that example. Suppose we want to model how EGF control cell cycle. We know by cell cycle, cells initially remain in G1, then enter into S phase, then G2 phase, then M phase where cell divide. This cell cycle, which controls division of cell, is activated by molecule, signaling molecule like EGF, epidermal growth factor. And we want to model that. Now from literature we know EGF eventually inhibits and activates multiple processes. And here in this particular case, we want to model how EGF controls a particular inhibitor P15. P15 is an inhibitor of cell cycle. So if you have to promote cell cycle, you have to inhibit that inhibitor, and EGF, that is epidermal growth factor, just does that. So here I have made a graphical model of that process. What is happening? The first step is EGF is binding to EGFR, the receptor, and they make a complex called EGF-EGFR complex, and obviously it is a binding process, so it is a reversible one. Once this complex has been formed, this complex trigger multiple processes. We have not shown all these processes. What happens after the activation of these multiple processes is that AKT, which is a kinase, get phosphorylated. So rather than showing all the processes involved in this, I have just shown an arrow, and here AKT is the substrate which becomes phosphorylated to PAKT. So this is the process by which AKT is getting phosphorylated to PAKT, whereas this complex EGF-EGFR is actually controlling this process. Once PAKT is formed, PAKT is the active form of AKT, and it is a kinase. It can phosphorylate FOXO to P-phospho. P-phoxo is nothing but phosphorylated form of FOXO, and P-phoxo is inactive. Interestingly, FOXO can control or activate expression of P15. So what is happening here? EGF is forming EGF-EGFR complex that is triggering phosphorylation of AKT to PAKT. Once PAKT is formed, PAKT is an active form. So that is an active enzyme that phosphorylate FOXO to P-FOXO. Now P-FOXO is not active, so transcription of P15 will decrease. So the whole process is represented by arrows and reversible arrows. Now once I have this graphical model, I will try to create mathematical equation to represent each of these processes. Just remember one issue here. This P1 and this P2 are phosphatases. So P1 is controlling dephosphorylation of PAKT to AKT, whereas P2 is another phosphatase and it is controlling dephosphorylation of P-FOXO to FOXO. Once you have this graphical model, we will move into making mathematical model. Remember, the thrust of this course is building dynamical models for biological processes or phenomena. By dynamical model means we want to model processes which are changing with time, which have a time dynamics. So we will use primarily ordinary differential equation. In ordinary differential equation, variables that we are measuring are changing with time. Sometime in some dynamical model, we have to use partial differential equation. Partial differential equations are used when you have more than one independent variable. So you have to use partial differential equation when you have more than one independent variable. For example, suppose you want to simulate movement of protein on a cell surface. So with time, the molecules are moving. So time is changing, also the position of the molecules are changing. So if you have some reaction happening on cell surface involving those protein, so with time, concentration of those molecules will also change. At the same time, position of those molecules will also change. That means I have to track or keep a record of those molecules with respect to time as well as space. So I cannot use ordinary differential equations in that case. I have to use partial differential equation. In this course, we will focus primarily on ordinary differential equation based model where time will be only the independent variable. That means the processes are such that we will keep track of changes with respect to time. I believe most of you are quite well acquainted with calculus, and in fact for this course we require +2 level school level calculus. We will recapitulate some basic concept of that, and I will also advise you, if you have forgotten some basic concepts of calculus, please look into the those basic concepts and try to recapitulate those basic concepts. By ordinary differential equation, what do you mean? See, ODE, a ordinary differential equation, is nothing but a derivative of a particular function. For example, here I have a function of t which is equal to x. X is equal to t square by 2 plus c. If you differentiate this one, x, with respect to t, you get dx dt, and by simple formula of differentiation, you get dx dt is equal to t. So this dx dt is equal to t is a differential equation, and it is an ordinary differential equation because here x is changing only with respect to time. X is the dependent variable because that is changing with time and it depends upon time, whereas time is independent variable. So we have a dependent variable x and we have a independent variable t, and together we have got a ODE dx dt is equal to t, that is the derivative of the function x equal to t square by 2 plus c. Now we will discuss about some basic difference between certain types of ODE. Let us start with the simplest one, the linear ODE. A linear ODE is such that the dependent variable and its derivatives have power 1 and there is no product of multiplication of its dependent variable and its derivative. Let us explain with this example. Here, this is a linear ODE. dx by dt is equal to a into x plus b. Remember, the dependent variable here is x. Its derivative is dx dt. The power of x is 1 here, and there is no power higher than 1 for dx dt also, and there is no product of x and dx dt. So that's why, as these two conditions are met, that the dependent variable and its derivative have power 1 and there is no product of the dependent variable and its derivative, this particular ODE, ordinary differential equation, is a linear ODE. I can have slight variation of this linear ODE also. Let us see that. See, in the previous example, in this one, a is a constant. Whereas in this equation, the second one, we have dx dt, a, which is function of time, into x, plus b, which is again a function of time. So b itself is a function, as a variable, dependent variable, it depends upon t, and a itself is a dependent variable and depends upon t and changes with time. But in this particular equation, we have only written the derivative of x, and we have no product of dx dt and x, and the power of x maximum is 1 and power of dx dt is also 1. So this equation is also a linear ODE. Let us see some nonlinear ODE. The concepts will be clear. The first one, dx dt is equal to 2x square plus 4. So the power of x, the dependent variable, is 2 greater than 1, so it is nonlinear. Look into this one, the second one, x dx by dt equal to 3x plus t. Power of x is 1, there is no issues, but we have a product of the derivative and the dependent variable of x, so it is a nonlinear ODE. Sometimes we differentiate different ODE based on its order. By order, we mean the value it is equal to the highest derivative of the equation. For example, take the first one, first order equation. Here the derivative is dx dt, whereas the second one is second order because we have d2x dt2. So this is second order derivative. In most of the problem that we will model in this course, we will deal with first order ODE. We may have both linear and non-linear. We will frequently meet situations where number of dependent variable is more than 1. In all the previous example, we have only one dependent variable, which is x. But you may have a situation, and that is much more realistic, and most of the biological problem you will have more than one dependent variable. You may have another dependent variable y. You may have a third one z. So x, y and z, all of them are dependent variable. So they are all dependent on t and they are changing with time. So for this type of system, we have to write separate derivative for each of these variables. So I will write one derivative for x, that means dx dt. I will write one separate variable for derivative for y, that means I will write dy dt, and I will also make a separate variable for the derivative for z. And if you see the example, the first ODE is for dx dt. This is my first ODE. Second ODE is for dy dt, and the third one is for dz dt. 3 dependent variable, 3 ODEs. Now, this system is called a set of ODEs. because I have more than one ODEs. One interesting thing here in this set of ODE is that notice that dx dt has y term, whereas dy dt has z term. That means if I have to know the derivative of dx dt, I have to know y, and that is controlled by this derivative again. Whereas dz dt also has x, that means I have to know about x to know dz dt. So x, y and z, they are changing with time and their values depend upon each other. So this type of system or set of ODE is called coupled system of ODEs. In most of the biological problem that we will deal with, we will have coupled systems of ODE. Now let us look into the utility of ODEs in our dynamical model. We have to remember that ordinary differential equations will be used to represent rate of processes. That is the utility of ODE in our mathematical process. So take an example. Suppose I have a chemical reaction. A is getting converted to B and the rate constant for that is K1. Whereas as it is reversible, B is also becoming A and the rate constant is K2. And I want to model this one. When I say I want to model this reversible process, essentially I want to write mathematical equation that will represent rate of change of B and rate of change of A. Let's see. So the first ODE that I have written, that represent rate of change of concentration of B with time. So how I am representing it with a ODE? I have written dB dt. Remember this square bracket represent molar concentration. So change in molar concentration of B with respect to time is given by dB dt is equal to K1 into A, that is the forward reaction, minus K2 into B, that is the reverse reaction. So B is formed by this forward reaction, B is converted back to A by this reverse reaction or backward reaction. So dB dt, rate of change of concentration of B with time, is equal to K1 into A minus K2 into B. Similarly, I have a ODE differential equation representing rate of change of concentration of A with time. So I have dA dt equal to minus K1A. Remember, by this process, this is the forward process, forward reaction, but by this process A is getting used up. So that's why I have a minus sign here, plus K2B, which is the backward reaction. And remember, here I have positive sign because by this process, this backward process, A is created. So actually both these ODE are exactly same except the signs. The signs are inverted. So these two equations is essentially my mathematical model for this reversible reaction. And the ODEs written here are representing rate of the processes. Rate of formation of B, rate of formation of A. When we are creating mathematical models using ODE, we have to make some basic assumptions. And those two assumptions should always meet, otherwise we cannot create a ODE based model. The first criteria is that the system has to be homogeneous or well mixed. Imagine a chemical reaction happening in a flask. A and B is reacting to make C. A plus B giving to C. If A and B are not well dispersed in the flask, if not well mixed in the flask, then somewhere concentration of A will be more, somewhere concentration B will be more. So production of C will be somewhere more and somewhere it will be less. That means the process will depend upon the position of A and B molecule inside the flask. We don't want to do that. So we have to assume, we have to make sure that A and B are very well mixed, so that I can imagine that the concentration of A and concentration of B are always same across the whole flask. So this is one basic requirement if you have to make a ODE based model, and you have to make sure that in reality that is true for your model. Second criteria is that the system size has to be very large. Otherwise, you cannot use ODE based model. Let me give you an example. Suppose I take a cell of volume, say 4 into 10 to the power minus 15 liter. A cell is usually 1 micron in diameter, so if you calculate the volume, it will come in this range. And suppose I have a molecule A, which is as a concentration of 10 to the power 10 nanomolar. If you use Avogadro number and use the volume of 4 into 10 to the minus 15 liter, 10 nanomolar means essentially 24 molecules. So inside a cell of 4 into 10 to the minus 15 liter volume, I have 24 molecules. So now suppose by this reaction A plus B giving rise to C, 1 molecule of A is used up. So that become 23 molecule. Now if you convert this 23 molecule in concentration term, it will become 9.58 nanomolar. So from 10 nanomolar, it becomes abruptly 9.58 nanomolar. So there is an abrupt change in the concentration just because one molecule has reacted to create C. Now imagine in the same volume, if I have 10 millimolar of A, that is equivalent to almost 24 into 10 to the power 6 molecules. So now if one molecule of A react, that becomes 2399999, and this is equivalent to 9.9999958 or similar to that. 58 or similar to that. So what I want to show here is that, see, if you have 10 millimolar, this is also millimolar, change in one copy of the molecule will cause a slight change in the concentration. So remember, here concentration change is not abrupt. If you have to model this system using an ordinary differential equation, the variable, the dependent variable should not change abruptly. They should be changing continuously. So for this type of system, if the concentration is higher at 10 millimolar, then I can easily assume that these changes are happening smoothly, continuously. But if the concentration is very low, suppose 10 nanomolar, then a simple change of one molecule will cause an abrupt change in concentration. And I cannot use ODE for this type of model systems. So the two key issues here, if we remember, is that we have to remember that the system has to be homogeneous or well mixed so that all molecules or all elements can interact with each other freely, and the size of the system, number of molecules, number of people, number of bacteria, that is all the elements involved in the process should be large in number. So let us briefly discuss one important issue in modeling biochemical reaction using ODE. Biochemical reaction can be very complex, but in some time we may use the law of mass action to simplify those processes. We all know law of mass action. If you don't remember, please try to recapitulate it from any biochemistry book or physical chemistry book. In essence, law of mass action says the rate of reaction is proportional to the product of molar concentration of reactants raised to power. So take an example. A is reacting with B. The small a and small b are stoichiometric constant and giving rise to M and N with a rate constant K1. So the rate of reaction should be proportional to the concentration of each of these reactant A and B raised to the power. So it is written as rate of reaction is equal to K1 into concentration of A to the power A prime into B, concentration of B to the power B prime. Remember, both these in square bracket are molar concentration. An individual rate, the rate of change of A is given by this. Rate of change of B is also equal to this, but its formulation will be different. dm dt is rate of change of M and dn dt is rate of change of N. And remember, each of them is divided by their stoichiometric constant. And notice the sign here. These are minus because there will be decrease in A and there will be decrease in B, whereas these are plus because M is increasing and N is increasing by this process. So whenever possible, we'll try to use this type of law of mass action equation. But remember, in all cases, you cannot do that. So let us jot down what we have learned in this module. If you have to create a mathematical model, first collect all relevant information. That is critical. Once you have collected all information, try to simplify. Keep the bare minimum essential thing. Then make some assumptions. And once you have made the assumption to simplify the model, create a graphical model. In all cases, you don't need it, but in many cases, it is better to draw a graphical model because it will help us to write down the mathematical models. Once you have done the graphical model, go for mathematical model. A mathematical model is nothing but a set of equations representing the processes. In this course, we will write ordinary differential equation to represent each of the processes. And remember, a ODE represents rate of a particular process. Remember, when I have to make a ODE based model, I have certain key assumptions. The first one is the system is homogeneous, that means well mixed, and the system is very large. That means number of molecules involved in the reactions are large in number. If I am modeling a cellular process involving multiple cells, so number of cells will be large in number. If I am modeling a process of a population of human being, number of human being in that population should be large in number. So these two key assumptions has to be met, and then only we can use ODE, create ODE based dynamic models. Thanks for watching. In the next modules, we will learn about creating simple models.